All right. We are here to talk about some fermentation. And we have quite a few. I just clumped a bunch. It was really hard to break up the fermentation I can statements. So 2, 4, 6, 7. There's 7 of them here together. But we're going to go through them fairly quickly, or at least I hope so. So the first one says I can differentiate between obligate and facultative anaerobes. So obligate, if you think about obligate, sounds like obligated. It is the same root word. So it means they are obligated or they have to do anaerobic respiration or fermentation. So these are organisms that can only survive, well, some of them can survive in the presence of oxygen, they just don't use it. Some of them, they can't even survive in the presence of oxygen. You're going to find them like deep in the mud where there's not a lot of oxygen, or underwater, or places where there just might not be a lot of oxygen available, because for some of them oxygen is poison. So those are your obligate anaerobes. The other is a facultative anaerobe, and that means that they can use anaerobic respiration as needed, but they also can do aerobic, and that's what we are. So we can flip back and forth. When we have plenty of oxygen, like right now, we're probably just sitting still. I highly doubt you're like on the elliptical or on the treadmill right now listening to this, watching this video. So you're probably just sitting there watching the video and you've got plenty of oxygen. And so you are doing aerobic respiration. You're doing glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation, and you're making those 34 to 38 ATP. If, however, you were to be under extreme circumstances, like you were working out really hard, where your cells are burning lots and lots of ATP because you're using a lot of energy to work out, but you're not breathing fast enough. I mean, when you work out, you do start to breathe faster, and that's because your cells need more oxygen. Because they're doing respiration faster, but at some point, if you're working out hard enough, your lungs can't keep up, and so your body, some cells are still getting some oxygen because you're still breathing, and they'll keep doing aerobic. But the cells that aren't getting the oxygen because you're just not bringing in enough for all of your cells are going to switch to lactic acid fermentation. And that's just a way for them to produce some ATP and stay alive until you calm down and start producing or start breathing normally. You know, you're not burning as much energy as much, and then your breathing will slow down, but you're at the point where oxygen is coming in at the rate that you need it. And so obligate anaerobes are those that have to do fermentation. They don't have an option. They can't do aerobic respiration. And then facultative are those like us who can bounce back and forth. So we can do whichever is needed based on our environment, based on our circumstances. That does make us a little more flexible when our environment changes. The more flexibility you have, the more likely you are as a species to survive. And so when you are an obligate anaerobe, and you get to a point where the only, for whatever reason you're taking in oxygen, you could die. So the fact that we're facultative and we can do both makes us a little more flexible, a little more likely to survive as a species. All right, I can explain the purpose of anaerobic respiration or fermentation. And the purpose of this is to keep producing a little bit of ATP. Now, fermentation does not produce any ATP. And you'll see this, and I'll talk about this again when we start outlining the process. But fermentation allows glycolysis to keep happening, so you get these 2 net ATP. So fermentation doesn't produce ATP, but it produces these NAD pluses so that glycolysis can continue. Because you can't do pyruvate oxidation, Krebs cycle, or citric acid cycle, or oxidative phosphorylation. You're not going to get any ATP that way, so you're relying on this ATP to stay alive. So you've got to keep doing glycolysis, okay? So the purpose of anaerobic respiration is to allow glycolysis to continue. All right, I can compare alcohol and lactic acid. So some ways that they are both similar. They both occur in the cytosol, okay? They are both done by bacteria and fungi, okay? Both of them, of course, do not use any atmospheric oxygen, so no O2. They both regenerate or make NAD+, which is why glycolysis keep going. They are both used to make food, anything from yogurt to sauerkraut, kimchi, alcohol and beer, bread, all kinds of stuff. And they both occur after glycolysis. So they do rely on the pyruvate from glycolysis to do their thing. All right, alcoholic is done, it produces, you start with pyruvate and you produce ethanol and carbon dioxide. And it is done by yeast, which is a fungi, a specific type of fungi. Whereas lactic acid is done by animals It's also done by other fungi and bacteria. So we've got bacteria and fungi here in the middle, but like I said, I don't want you to think I'm thinking this is not a fungi. This is just a specific fungi that does alcohol fermentation. And it's going to produce lactic acid. So a lot more similarities than differences for our purposes, but there are a few differences, and the product is the main difference. Okay, so that's comparing and contrasting. Then I can outline the process of fermentation. We're actually going to outline both of them. So we're going to start, and we're going to do them in 2 different colors to keep them straight in your brain, in your little noggin. So the first one I'm going to do is going to be alcohol. Just because it's on the left up here, I'm going to put it on the left down here. Okay? And so alcohol fermentation takes that pyruvate, and it is going to convert it to ethanol, which is a 2-carbon. If you'll remember up here, pyruvate's a 3-carbon. And so it is going to lose a carbon dioxide. Now, it is not aerobic. I know in citric acid cycle we lost a carbon dioxide and it was aerobic because atmospheric oxygen came in and picked up that carbon. That's not the case here. These oxygens are coming off of this pyruvate. So this C and both of these O's are coming from here, and that's why it's not aerobic. It is anaerobic. When that happens, or when the conversion of pyruvate to ethanol is going to need the addition of some electrons. So what we're going to do is we're going to take these NADHs produced in glycolysis, and we are going to strip off their electrons and give them to pyruvate to make ethanol. So pyruvate is going to be reduced because it's going to get the electrons as it becomes ethanol, and your NADH is going to be oxidized into NAD+. Now this NAD+ can then go back up here to be reused by glycolysis, so glycolysis can continue. So that's what you would do if you were a bacteria or a yeast, certain bacteria and yeast. What we do, what we and some other organisms, some bacteria and fungi, is we take pyruvate, which again is a 3-carbon, and we convert it to lactic acid, which is also a 3-carbon. So we're not losing any CO2s here, but that conversion is going to need some electrons. So what we're going to do is, again, we're going to take this NADH, we're going to remove the electrons. Okay, so we're going to oxidize it back to NAD+, which means we are reducing the pyruvate, making it lactic acid. And then that NAD+ can now go back up here and be used by glycolysis. So you'll notice there's no ATP made in either one of these. See, no ATP. But what it's allowing is it's allowing us to continue doing glycolysis, because if we just did glycolysis and we didn't have fermentation, we wouldn't have any NAD+. All of our NAD+ would be made into NADH, and then glycolysis couldn't continue. You got to have NAD+ for that to continue. So this is a way for us to convert that NADH back to NAD+, so glycolysis can continue. So that is outlining the process. The reactants, I would say the reactants over here would be your pyruvate, pyruvate, your NADH, and that's probably it. And then your products would be ethanol, CO2, and NAD+. Okay, on this side, your reactants are pyruvate and NADH, and your products are lactic acid, which sometimes you'll see that called lactate. It's the same thing for our purposes. There is a slight chemical difference, but it's the same pretty much molecule, and NAD+. Okay, so those are, I can outline the process, make sure you can do both, and I can list the reactants of fermentation, and I can list the products of fermentation. That also goes more also into describing the purpose of anaerobic respiration or fermentation, which was one of our I can statements that I mentioned a second ago. So I hope that helps give you a better feel for respiration, and maybe, hopefully, you can do great things with that concept right now. And then I think that's it. So if you have any questions, let me know.